Science is my work, poetry is my inspiration, and humanity is my ideology. Science is my fortune, poetry is my happiness, and humanity is my peace. Science is my sense, poetry is my mind, and humanity my heart. Science is my friend, poetry is my heart, and humanity is my God. I love science, respect poetry, and worship humanity. On this note, I, Shadma, take immense pleasure to welcome all the honorable dignitaries, invited experts, esteemed guests and delegates, deans and heads of various departments, faculty members, media persons, and dear participants. On behalf of Department of Biotechnology, MNNIT, Allahabad, for the 5th version of the repeated conference series, BioSangam 2022, an international conference on emerging trends in biotechnology. BioSangam 2022 is aimed to provide deep insights into innovations, challenges, and growth opportunities in diversified domains of biotechnology. With a plethora of sessions by eminent oracles lined up for the next 3 days, it consists of various sessions including keynote, plenary, invited, and parallel sessions. Each session will be addressed by outstanding experts who will be providing a forum to researchers around the globe to explore and discuss on various aspects on recent advances in the field of environment, food, agriculture, healthcare, and pharmaceutical, interdisciplinary research, design, innovations in IPR that has dynamically opened up new avenues of research. It will provide a deep insight into innovations, challenges, and growth opportunities in diversified domains of biotechnology. To begin this auspicious occasion with the blessing of the Almighty God, I request Chief Guest and Honorable Director, Professor Ramashankar Verma, sir, Guest of Honor, Professor P. Nagabhushan, sir, Chairman BioSangam, Dr. Vishnu Agarwal, and Treasurer BioSangam, Dr. Manisha Sachan, ma'am, to please come forward and light the lamp as a symbol of imparting knowledge and enlightenment and take their seats on the dais. Please welcome, sir. Please welcome, ma'am.
कुंदेन्दु तुषार हार धवला या शुभ्र वस्त्रावृता या वीणावर दंड मंडित करा या श्वेत पद्मासना या ब्रह्माच्युत शंकरा प्रभृतिभिः देवै सदा वन्दिता सा मां पातु सरस्वती भगवती निशेष जाड्यापहा शुक्लां ब्रह्म विचार सार परमा आद्यां जगद्व्यापिनी वीणां पुस्तकधारिणीम् अभयदाम् जाड्यान्धकारापहा जाड्यान्धकारापहा जाड्यान्धकारापहा.
A very warm welcome to all our dignitaries on a stage. Can you please have a huge round of applause for all of them? Thank you. I would like to Smriti to please come forward and welcome Director R.S. Verma, sir, with a bouquet of flowers. I would like to request Padmaja to please greet Professor P. Nagabhushan, sir, with a bouquet of flowers. Shubhangi to greet Chairman BioSangam 2022, sir, with a bouquet of flowers. I request Disha to please greet Treasurer BioSangam 2022, Dr. Manisha Sachan, ma'am, with a bouquet of flowers. I would like to head of the department and Chairman BioSangam for his welcome address.
Very good morning to all. Honorable Director, Professor R.S. Verma, sir. Of the departments, deans, faculty colleagues, and now live streaming eminent speakers, students, and guests. I am delighted to welcome all participants to the BioSangam 2022, Emerging Trends in Biotechnology. It is a unique conference to provide a platform to faculty, scientists, researchers, students, and learners to come across new eras and emerging trends in biotechnology. This BioSangam 2022 is the 5th edition of the conference, and since its first edition in November 2013, this conference has gradually gained its fame and now the most awaited conference in the region. Motilal Nehru National Institute of Technology, Allahabad, Prayagraj, is an institute with total commitment to quality and excellence in academic pursuits. It was established as one of the 17 regional engineering colleges of India in year 1961 as a joint venture between the Government of India and Government of Uttar Pradesh. On June 26, 2002, MNNIT was transformed into National Institute of Technology, fully funded by Government of India. Moreover, in year 2007, with the National Institute of Technology Act 2007, the institute was granted the status of Institute of National Importance. The Department of Biotechnology is going to organize the BioSangam 2022. The Department of Biotechnology at MNNIT Allahabad, Prayagraj was established in year 2006 to integrate the life sciences with engineering to develop cutting-edge technology through research training, technological innovation. The department is running BTech, MTech, and PhD programs, and the department has young, enthusiastic, and well-qualified faculty members actively involved in teaching and research. The name BioSangam has a broader holistic meaning. It means that meeting different peoples and thoughts in a single place, like a holy river Ganga and Yamuna meets in Prayagraj city and creates Sangam. This COVID year, due to COVID-19, the conference is being commenced in online, but the positive point is that participants may get connected from their respective places without exhaustive and expensive travel. Different interactive sessions like keynote, plenary, and invited talk shall enrich the participants' knowledge. Students from all over India are participating in the conference and connected with distinguished scientists and researchers worldwide. Different awards category for the poster, oral, and young scientist awards shall be given to the student to motivate their hard work and dedication. The event success results from tireless efforts of the person working behind the curtains. And I extend my gratitude to all my colleagues and whole team for their efforts and support. Again, I welcome all the delegates and participants, and I hope this conference will be a grand success. Thank you very much.
Thank you very much, sir, for your kind words. We also have with us Professor P. Nagabhushan, Guest of Honor, Director, Indian Institute of Information Technology, Allahabad, is Professor in Computer Science and Engineering from University of Mysore. He has more than 3.5 decades of teaching experience. Recently, he has been awarded Lifetime Achievement Award for Outstanding Achievement, Global Leadership, and dedicated service to the field of image processing and pattern recognition. I would like to invite Sir for his valuable remarks. Please come, sir.
So good morning to all of you. Let me at the outset welcome your new director, Professor R.S. Verma, to Allahabad, because I happen to be an older guy. So let me take the pleasure. But ironically, Professor R.S. Verma is from Uttar Pradesh. I am not from Uttar Pradesh. Look at the scenario that I am welcoming him as a UP man. So that is the integration. Possibly that is the other name for Sangam. That is the other name for Sangam. Sangam is Indian always. I was really happy. I was very pleasant. It was a very pleasant evening. And management. I was really wondering where did this come in? In fact, people always do that. They manage the papers, they manage the H index, they manage the I-10 index, they manage citations. They write the papers to manage producing a paper. Probably is the management so for that? I do not know. But then I really liked the poetry being added to science, because the rhythm of science is always in the poetry, or the rhythm of poetry is in the science. For instance, you just listen to this one small poem. This is the poem where the princess falls in love with a soldier, and this is the time the exchange of their talk is going on. The poet writes like this: love swells like Salve. Salve is the name of the river. Love swells like Salve, but ebbs like its tide. What a wonderful mathematics in that. Love swells like Salve, exponential rise. Always it does happen. You know, this lady is nodding the head. I think she must be having a lot of experience. love swells like Salve. Okay. The second part is probably true to all of us. Ebbs like its tide. It ebbs like its tide. So wonderful exponential rise and an exponential decay be through the literature. Ebbs like its tide. Wondering why, starting from edition, the BioSangam has been so strongly remaining. Naturally, after 3 years, and particularly in a situation where there is rotation amongst the heads is common, everybody next head always should make a U-turn, and always the next person should give a new name to the international conference. But still, the name BioSangam has remained invariant even during the 5th edition. I was wondering what has got to be the cause for that. Because that is the philosophy. While she narrated the Sangam of science, poetry and management, I would love to mention this Sangam as a wonderful confluence of science, the bioscience, with engineering. It is basically the confluence of engineering and bioscience, as Dr. Vishnu was trying to explain at the outset. But then there is invisible Saraswati. I do not consider, particularly I do not consider mathematics as either science or engineering, because mathematics is the backbone. And therefore, the invisible Saraswati in this confluence is the mathematics. Therefore, this Sangam is perfectly a wonderful confluence of, has got to be the confluence of engineering, science and mathematics. And I am very sure you are the right person for your department, Professor R.S. Verma himself. And from science to technology, his migration has been so wonderful, and I'm very sure he would be the right chief guest also for the occasion. While explaining the science, the bioscience, your bioscience itself is a confluence of many application areas. Pharmaceutical, medical, lot more. Zoology, lot more. It's a very wonderful area. Then, naturally, engineering. It is not to offend anybody. One has got to clearly understand where science starts with a statement of a hypothesis, makes it a theorem, where mathematics takes along to prove that theorem, and finally where science and technology would put a QED, you know, that quad era demonstrated, QED, there the engineering commences. So when the formal proof is over, when everything is over, the engineering commences. Where the confluence of science, mathematics, and engineering will transform an idea into a product, into a reality, and from there the reality, how far will it be useful to the society is what has got to be measured, and its effect on the society which has got to be measured, not the H index, not the citation, because after all, even a big international conference like this can have only so many people, and even among so many people, only few would read your article, only very few would be interested in your article, and asking a stupid question like, how popular is this paper going to be, is as ridiculous and as stupid as the question itself. I really cannot understand why editors would always ask this very nonsense question that how effective would this paper be, or how popular would this paper be. Effectiveness and popularity cannot and shall not go together, because effectiveness is triggering a co-investigator on the importance of the paper, whereas the popularity of the paper will be realized only when that paper becomes a product and that product becomes applicable to the entire society. When society accepts the idea late, not the idea, the product later, which has happened because of that, then it is popularity. And popularity cannot be measured when the paper is submitted for the review. It can be projected that it has got a strong potential. So people have got to understand. What I am trying to tell you is research. Let us be researchers, not for namesake, but for the depth that the research requires, but for the satisfaction that we derive, but because it is a passion for me that I want to do something. That has got to be there. I am very sure the young researchers here as well online would certainly be motivated that research is not just to get a PhD degree, research is not just to get a postdoctoral degree, research is not just to get a position like assistant professor, and research is not just to move along to become an associate professor, and finally trying to become a professor is not the goal, but it is a passion. When you do a research, you do it not expecting that you are going to become a higher scientist, a bigger scientist. You do it because you are going to make a contribution which leaves a wonderful, wonderful mark wherever it goes. You are not doing it that with the intention that I want to do such a research which leaves an impression. You are doing it because you are finding a way. You are doing it because you are trying to see the light at the end of the tunnel, and you want to explore that light at the end of the tunnel. You want to experience that light because you want to touch that light as a first person in the tunnel. That is all. That has got to be the spirit of research. I'm very sure, unfortunately, though the researchers are supposed to be the great philosophers, they have got to be. That is why the degree is called PhD, where it is not mentioned as even MTech degree connotes that it is MTech in technology, whereas PhD does not mention that. All right, PhD has no bar at all. It is philosophy. It is doctorate philosophy. Right. So when we speak about philosophy, unfortunately, we people, though we speak about philosophy, we also staying back in yesterday or jumping into uncreated tomorrow. The tomorrow has still not happened. Yesterday is gone. Both are not in our hands, and we still feel that both of them are in our pocket. We still feel that both of them are in our control. This is the worst part of it, and this is possibly be the worst for any researcher, because today is mine and today is the day for my research, and today's change would definitely be hopeful for a tomorrow. and would possibly make an yesterday's dream a wonderful, glory. That's all. So neither yesterday nor tomorrow is in our hand. Today. We shall be worried about today. Today is mine. Tomorrow you may become a professor. Day after tomorrow you become a director. Day after tomorrow you may become something else. Who knew Ukraine war would be there? Absolutely. People were talking that there shall not be any war at all in this world. But there are people, there are lunatic reasons for these things to happen, and the whole world is all the time is going to be stochastic. But then, today, which is in my hand, is a real reality. We shall live today. We shall enjoy it. Because I slided into philosophy, towards the end, let me just share only two things which we have got to remember, two things which we have got to forget in our life. The two things which we should forget are the following: the good that I have done. The good that I have done. Let us also forget the bad others might have done to me. The good that I have done to others, let us forget. And the bad others have done to me, let me forget, and which I always keep doing, because I want to forget about whatever the bad things done by my wife to me. So I always do that. Always forget the good that I have done to my wife. Always forget the bad that she could have done to me. Conversely, you just remember two things: the good others have done to you, and you should always remember, most important, the bad that you have done to others when your own consciousness comes to know that you have done bad to others. Because that is where science springs. That is where math gives a foundation. That is where engineering blossoms the science into a product. Thank you very much for such a patient listening.
Thank you so much, sir, for your kind and inspiring words. We are honored to have you here. We have among us Professor R.S. Verma, sir, Chief Guest and Honorable Director, MNNIT Allahabad, former head of the Laboratory of Stem Cell and Molecular Biology, Department of Biotechnology, IIT Madras. Sir is a postdoctorate from Division of Oncology, School of Medicine, Indiana University Medical Center, USA, and NIH, USA. He is a recipient of several prestigious awards, such as Fellow of National Academy of Sciences, Applied Biotechnology and Applied Pharmacy, Senior Scientist Award, Medical Biotechnology Award, and Fellow of Society of Applied Biotechnology, as well as Fellow of Association of Microbiologists of India. I would like to take the privilege to invite Director Sir for his presidential address. Please welcome, sir.
Thank you very much. First off, I would like to welcome Professor Nagabhushan for our BioSangam 2020 and his inspiring talk. And he has set a tone for this BioSangam. I think everybody will agree with me. He started with the poetry and then he went over the philosophy. I think philosophy of the science is very different. I would say is the question of understanding a small thing is basically is philosophy. And how we can achieve in research, when we do something, we leave legacy of questions to be answered by others. And that should be the way of a researcher, because any research you do it, you cannot fulfill all the answers. You have to answer only one at a time. And then when you answer this question, the another question pops up from that work. I think that's where the whole science goes around. And he's rightly said, until you deliver any product, it will not be popular, which is very, very, very important. So what you do, not understanding that it will be finally go to the market, but do something in that direction. I'm sure thousands of people do the research, only one or two product goes to the market, not all of them. But it's a collective research and information which they share in the research. I think that helps us to do that in that one. As he said, he has welcomed me from outsider, and I'm really thankful to our fate, our MNNIT, who gave me chance to lead this institution. And as I told you that I'm a bench worker. I do work on bench and talk to my students a lot to have the question, because I give importance to the students' thought. That's where I progressed. I never impose my way of philosophy, but yes, I will make sure what they're thinking, I'll polish their thinking during the talking. So this BioSangam is really is a nice idea, and I can share with you, I think I came once also in this BioSangam earlier to give a talk also, but that was at least, I don't know, when he came, 10 years back, something like that? Huh? 8 years back, okay, so I'm close. So at that time I didn't know that I will come as a director here, to be honestly, you know. Even I did not know a few days before I was leaving to US that I'll be coming back here. And I tell you, as he said, I'm from UP, yes, I am from UP, but I'm really more nationalistic. Why I'm saying, if you see in Jangan Mangan, so there is a place there, Punjab, Sindh, Gujarat, Maratha, Dravid, Utkal, Banga. So I started at Punjab, Sindh is not there, then I went to Gujarat, then I went to Maharashtra, then I went to Dravid, Utkal, Banga I didn't go, I came back to my whole thing. So it's a whole circle came to me actually, and I'm very happy to do that. Let's talk about a little bit of biotechnology. Biotechnology is basically emerged as a different discipline in Department of Science and Technology in 2003, basically. No, it was in 1983. Okay, 83 it came as a distinct area of a research where the biologist, which was headed by basically a botanist at that time, and some of the genetics, who came this idea that this discipline has to be nurtured. And since 1983 onwards, it was a small office in IIT Delhi that started this concept of biotechnology, and now within approximately 40 years roughly, it has come in the height of Indian science. We have done so many institutions right now, opened by the Department of Biotechnology, covering a discipline of plants to higher animals also. So they have contributed a lot to understanding the basic sciences versus some of the product development also in that one. And I think next, and you say all this technology which has been in India and came from the people who came, taught, they got a kind of experience in abroad, they came, and during the COVID period, the 2 vaccines were developed by those biotechnologists basically. And this technology was a forefront in whole world, which we have made it. I think this has creating a plethora of the Indian science to be recognized by all over the world, because we have not only saved our Indian population, but we have saved and to other country population also. In that regard, we have supplied a lot of vaccine to them. So this is one example that it has gone to the popularization the biotechnology product. One product basically, but there are biosimilars are there, which in the market. Reverse genetic engineering is there right now. DNA fingerprinting was one of the area which is started by studying only the snake venom of different in BHU by Professor Lalji Singh. And today's article also came in some of the newspaper if you read his, and he has established institution also. And now the DNA fingerprinting, which is a part of the genetics, has become a technology. So you can see how the small thing which we have studies in BScs, in intermediate kind of things, they have now a technological things which is more applicable. So this conference is really well done, and it is the covering different parts, different areas will help to the students. And I would request all the students that they should not do the experiment just for getting the PhD, what he said correctly, Professor Nagbhushan, that you understand the philosophy of the science, philosophy of doing the work. If you answer only one questions in your whole PhD, I think that is much better. You should not go and do small work just to get the published paper, what he said correctly. And I will make sure that those people who publish in a higher, good quality work, they will be recognized. It is not only numbers, it's the quality of the work which is recognized. It will popularize soon. It may not popularize in one year. A lot of people, if you see in IIT also, IIT setup, the people don't publish paper in a quality journal. Number is more important because this is a race for becoming a associate professor and professor. This is one thing which Indian science is lagging behind. I mean, it's taking back basically the innovations. Innovation does not come with the publishing a paper in 1 year, 10 papers, 20 papers. I mean, I remember there is one person from chemistry department, I think he published 100 paper in one year in IIT Madras. 100 paper, I'm telling you. And he got promoted from assistant to professor immediately, within 2 year point. But after that, his productivity is not recognized at all. Because people understood that what he has done only the numbers. Maybe few papers are quality, but this quality work has been recognized basically. So the number is not important, it is the quality of the work important. The popularization will happen later on. We don't have to worry. I can share with you in Oxford, University of Oxford, in Cambridge University. The people retire as a reader, but they got Nobel Prize also. They don't worry about the promotions. They have dedicated their life to understand the problems. Our Indian, what is his name? Ramakrishnan. He's from Baroda, basically. He's basically a physics graduate. He did PhD in physics, then he moved to the biological science, and he moved from US to Cambridge. If you meet him, you will never see that he's a Nobel laureate. He's always busy in solving some questions in his laboratory. So I think those people are icon. If you meet them, to live with them for 1 or 2 days, really influence a lot. So probably Professor Nagabhushan has given the tone for this conference. Maybe next time you should ask somebody, to come a real hybrid person who has done something interdisciplinary, and get them, and ask them to spend at least 1 week here, interact with the people of different discipline, not only the biotechnology, there's technology other part also. People will not like it, but I like biomaterial much better. Because material science gives you the base for making any tissues, to understand the whole things, and I'm very fascinating about the material science. See, earth is a material, right? Everything comes from the earth. Think about how earth has given the, everything to you, but you have to use it. You have to make use of that particular things. From the energy, from minerals, from the food, right? Everything is given by earth. So how a earth can sustain such a long period of time giving all the things to you? We must understand those things. I think he is also very much touched about the science and engineering. So I think in the future we should call engineering science. One discipline has to be there. Not only the science engineering or engineering science kind of things. See, traditional subjects are gone these days. Interdisciplinary subjects have come, because we have to, not only in one area, but in a different area which, help each other. That's important things. I will not take much time. I thank you for organizing a beautiful conference, and all the colleagues in Biotechnology department. I think yesterday I got appointment in biotechnology also. So I'll be sitting there also sometime. And I'm making sure that they come on time. Because when you are in the lab, you will work. If you don't come, you will never work. This is my philosophy. Thank you very much.
Thank you so much, sir, for your very inspiring words. Now I request all the dignitaries in the dais to please release the souvenir. Thank you very much. We are all aware that BioSangam 2022 was to be held at one of the most ancient, holy and culturally rich city of India, the KumbhNagari, Prayagraj. But unfortunately, due to the ongoing COVID-19 situation, this conference is to be held in the online mode. Hence, we have a screening of a short video that will help you in exploring our city, that will introduce you to the spiritual, divine, ancient, historic, archaeological, as well as the political importance of Prayagraj. So we'll be having a small screening of a short video. video from our side so that we could show you all how beautiful our city is, as well as how green and beautiful our campus is. So I appreciate that. On this note, now I would like to request, we will be starting with the memento presentations, with the felicitation ceremony. So I would like to request Dr. Vishnu Agarwal sir to please present a memento to Director R.S. Verma sir as a mark of reverence. Can we have a huge round of applause, please? I would like to request Dr. Manisha Sachan ma'am to please present a memento to Professor Nagabhushan sir as an expression of our love and respect. Thank you very much. Now I would like to request Dr. Manisha Sachan ma'am to please deliver the vote of thanks.
Chief guest of this inaugural session, Professor R.S. Verma. Guest of honor, Professor Nagabhushan. Chairperson of the event, Dr. Vishnu Agarwal. Heads and deans, faculty members, participants, and students. Good morning. I deemed it a great honor and privilege to propose vote of thanks for the inaugural session of this international conference on emerging trends in biotechnology, BioSangam 2022. First and foremost, on behalf of the organizing committee, I would like to extend my heartfelt gratitude towards, first and foremost, towards Professor R.S. Verma sir, who is director of our institute and patron of the conference. This conference would not have been possible without his support and encouragement. Professor Verma has a very significant contribution and vast experience in the field of biotechnology, especially in the domain of stem cells and molecular biology. We are obliged to have you, sir, as chief guest of this occasion. I would like to express my wholehearted sense of thankfulness to our guest of honor, Professor Nagabhushan, sir, who is director of IIIT Allahabad, who has spared time from his busy schedule and enlightened the budding minds. The organizers are highly gratified towards heads, deans, advisory committee members, and other institute functionalities to make this event a grand success. My heartfelt thanks to all plenary and invited speakers and to the judges of different events for their contribution and taking out time to share their insight and wisdom to modulate young minds. I owe special gratitude to the faculty in charge of Center of Continuing Education, Professor G.P. Sahu and Dr. Manish Tiwari for providing leeway for the organization of this conference. Also, we are extremely grateful for the support provided by Civil and Electrical Maintenance section of the Institute. Sincere thanks to the in charge of Computer Center for uninterrupted internet facility. I would also like to thank the EDC in charge and staff to extend their services for the lunch. Also, I thank to Atlantis Press for accepting full-length manuscripts to be published in the Scopus Index Journal after completing the reviewing process. I thank all the sponsors for their support. The actual brainstorming happens when the organizing secretary, along with the organizing committee members, delves into all aspects of the event. Therefore, I express my heartfelt gratitude to the chairperson of the event and head of the department, Dr. Vishnu Agarwal, and the members of organizing committee for their untiring work. I thank all the distinguished invitees present here, media person and participants enrolled for this session. Last but not the least, the force of action of the event, our student volunteers. So I would like to thank all the student coordinators and volunteers for their persistent efforts. Once again, I thank you all for your presence here. Thank you.
Thank you so much, ma'am. Now I request everyone to please rise for the national anthem.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तब शुभ नामे जागे तब शुभ आशीष मांगे गाहे तब जय गाथा जनगण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
To the end of our inaugural session, I request all of you to please proceed for high tea. But before that, there is a little announcement. The keynote lecture will be beginning at 11:45 a.m. So all the guests and the participants are requested to kindly join 5 minutes prior. Thank you very much for your presence over here. Thank you all. You all may proceed.
hello, 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 hello. Yes, yes or no?
School of Medicine, Indiana University Medical Center, USA, and NIH, USA. Sir has years of academic experience as professor at Department of Biotechnology, IIT Madras, adjunct professor at Faculty of Science, Engineering and Technology, Swinburne University of Technology, associate professor at Department of Biotechnology, IIT Madras, research assistant professor at Department of Medicine, Indiana University, associate professor at Department of Biotechnology, NIPER. Sir also has industrial experience as head at Pharmacology Division, Lupin, and general manager at Department of Biotechnology, Cadila Healthcare Limited. He is a recipient of several prestigious awards such as FNA, Applied Biotechnology and Applied Pharmacy Senior Scientist Award 2014, Medical Biotechnology Award and Fellow of Society for Applied Biotechnology 2011, and Fellow of Association of Microbiologists of India 2010. Sir's area of research include transdifferentiation study of stem cell in cardiac tissue on genetic and epigenetic level, study on 3D culture of stem and cancer cells under microgravity, construction of novel cytolytic fusion proteins against cancer, drug delivery system against leukemia, and liver and heart valve tissue engineering. He has guided more than 32 PhDs and 25 MTech and MS students and published books, more than 180 research papers, several book chapters and review papers on stem cell biology, tissue engineering, immunotoxin-based cancer therapy, papers in healthcare material, cancer letter, apoptosis, nanomedicine, etc. Sir is a reviewer for DBT Wellcome Trust, DBT, DST, BRNS, ICMR, and DRDO funding agencies. and a member of Biosafety Committee, Institute of Ethics Committee, and Animal Ethical Committee. Member of Board of Governor, National Institute of Pharmaceutical Education and Research, NIPA Mohali, and a member, Stem Cell Committee, nominated by the President of India. I request, sir, to please begin the session on the topic of fabrication and in vivo assessment of cardiac patch and injectable hydrogel for cardiac tissue restoration in myocardial infarction, rat model. Please welcome, sir.
So, I am going to speak, can you hear me all? Speak basically a recent work which my as a third PhD student had performed, Vinita, and this is actually a 3-part presentation. Basically fabrications and assessment of cardiac patches, and then their injectable hydrogels for cardiac restoration, and also a infarcted model which I am going to talk about. This was a extended studies of my cardiac nanogels which we prepared in the cardiac tissues. I will tell you how to do this basically, and also using some herbal product to deliver the proteins or components which can enhance the cardiac proliferation from stem cells. So, these are 2 talks basically I have combined together, and I will take around 35 minutes to finish my things for you guys. I will not take more time. So, as you know, just a brief introduction of the global side. It is old data. Approximately 18 million people die every year worldwide. Estimated that of the total 31% population die because of the cardiac arrest or cardiac related diseases, and also 85% of cardiovascular disease basically contains the heart attack and strokes basically. And you can see here India is one of them in this one, which is approximately 500 per million population gets the heart attack or stroke in lifetime here. That is the Indian statistics basically. So, what is basically, this is a normal heart and this is basically injured heart, which is a myocardial. So, the myocardium is basically a name for the cardiac tissues, which is not a skeleton tissue, a different kind of tissue because it has a beating properties. Other muscles do not have a beating property. So, that is what is a cardiac, basically known as the myocardial, okay, because the structure of these skeletal muscles are very different than the other muscles in this regard. And this is all structure, there is the aorta, pumps, and so there are 2 ventricles. This is basically ventricle side where some of the tissues get damaged with the time, because what happened in tissue, the blood supply, arteries which supplies the blood to these areas, they get blocked basically. So, there is not enough oxygen in that particular region of the myocardium, and they get in blacks. So, they lose their ability to beat in that particular. So, what happened when they beat the ability, these basically the heart pumps in very strong, because some of the, because the work is distributed to other tissues, and they try to squeeze the blood to pump into the lungs for purification. That is why you get the pain basically in that particular moment. So, there are so many diseases are there, I mean drugs are there, intervention. So, what is the treatment for this kind is a beta blocker, ACE inhibitors, and bypass surgery. So, bypass surgery means that you cut the region which is blocked and then bypass, make another artery or take from the other places and just bypass those things. That is what the bypass surgery is there. There 98% people get relief by this treatment, bypass surgery basically, but there is a recurrence is there that another artery also get blocked because our food habits are very different. That is what this is keep happening. So, ultimately what happened basically, surgery is there, then again bypass is required after some time. There is another things also use the stent. So what the stents basically, they will put some metal ring or thing, so we can increase the dilation, the part of the artery, so the blood can flow. But again it is blocked after some time, maybe after 5 years, 10 years, depending upon food habits basically. So limitation is that this you require a long term medicine, and several side effects are there with this kind of things. So cell therapy is the last 10 years, I would say that the people have tried that how to revive this infarcted region. Is there a possibility? They try to inject on the site these stem cells and see the stem cells are different kind basically. I am not going into detail of those things, I can talk little bit during my usual classes, what are the, and you can use it. So and then after, how to deliver those cells which can transform into the cardiac tissues with the right orientation. See, important thing is there, if they make a cardiac tissues, if not beating synchronously, then this will reverse effect will happen. So you have to be careful when they are making these tissues, it has to be right direction beating. So the cell therapy also is a good idea, but there are two things are there. Low retention, because of the beating property, if the cells are going there, continuous beating, cells come out, they do not stuck to the place, or they do not stay their place where they are supposed to be there. Okay, so that is a big problem. Second thing is poor engraftment after donor cells, because donor cell is nothing but the cells basically. So engraftment means that you are putting the cells in the place, it has to be there only. If they are moving somewhere else, they will not perform the function. And then these are the undifferentiated cells, they have to differentiate into the cardiac cells. So that multi-ways problem is there. First retention, first delivery basically, then retention, then basically differentiation, the proper orientation. So you have to address all the issues in that one. So to scar burden is my treatment is a critical problem in the clinics basically. And then what we are going to do that, we are going to do is that here the cardiac tissue engineering strategies combining scaffold. So how do you put the cells into the proper place? So we are using a scaffold here basically, and then we are having to have bioadhesive, because cell has to adhere there, you require some glue, otherwise they will come out. So first of all you have to develop a delivery system, then develop basically a molecule which can attach the cells, and third thing is that the cell which you are putting in that gel, it has to be cross that barrier into the tissue. That is important. So you have to address all these tissues, all these questions. So basically overview of my work is there in this one. So what is basically here, the potential of decellularized material. Decellularized means that you take a cell and remove all the cellular component, but the scaffold is left, the architect of that particular cells should be left behind. So you develop a strategy in such a way that you not destroy the architect, and this is a very trivial, very tricky proposition. You can't do that without destroying some architect. So once you have a decellularized matrix, we have used this basically a cardiac cell. So we have isolated the, well, I'll explain you later on. We take the cardiac cells from the heart, okay, and we decellularize them. So we know that there is an architect is around, and then we try to use as a scaffold, because what happened that if you put the similar kind of scaffold into the tissue, acceptance rate is much better, because they will say that this is our own, they will not from other tissues, because there are a lot of organic and inorganic other materials scaffolds are available. So we have not used that one. Second problem is that suturing the things, that when you make a scaffold, how do you suture the place of infarcted region? So that question you have to address. So either you sew it with the needle, or you do some bioadhesive. So we have developed a bioadhesive kind of things. I'll tell you about that also. And then this is basically characterization of those things. And then finally what we did basically, because we have to put certain enhancer molecules to develop these normal stem cell into the cardiac cells. So you need some promoters. And promoters is not only the protein, other things also you can use, chemicals also. Here we use a 5-azacitidine, which is a known drug for converting stem cell into the cardiac cells. This has a proven basically. So, I will focus on these three things basically. So, here is our hypothesis that this exploring all those things I have told about that main thing is that cardiac tissue restores functionally, we have to restore the cardiac function. Whatever it is the final outcome, that is what say, whatever deliverable is there, that you make this heart again into the same pace of beating. That is main idea. So, to explore the applicability of these biomaterial for regenerative therapy throughout in vitro and vivo stage, I will show you some of the work which we have performed in this area. So, now the first thing is that this is a cardiogel which we have isolated and develop a procedure for how to decellularize the things, and we make a gel out of that. It's basically a sticky, it's a scaffold which is sticky. It's sticky in the sense it has a lot of water content, it's a hydrophilic, so that the cells can go inside. That's an important thing in that one. Fabrication of the engineered biomimetic cardiogel patches and in vitro assessment, because this is, and the development of injectable, these are two things we will talk about. So, this is the basically cardiac gel. What we did, we have taken a neonatal mice. So, this is the two things, you can take adult rat also, adult mice also. But what happened when the tissue is differentiated finally, then it's become very strict to convert back to those things, the normal things. So, and then the tissue is very hard. If you grown tissue you take and try to decellularize, it's very difficult to decellularize because it has a lot of layers of scaffold in that one. And where is that basically in this one? So, what we have taken neonatal mice, which is basically a 1-day-old mice after delivery, and then we have taken a heart. So, this is a neonatal mice, and then we have crushed it with the scissors, scalpel basically. We have crushed it and then we have plated on the cell, and then we did lot of subsequent massages and used lot of material to decellularize them. So, that is what we did basically. So, decellularization process has been done. After decellularization we call them as a cardiogel, because a gel which is extracted from the cardiac tissue, that is what we name a cardiac gel. This paper we have published in 2012 in PLOS ONE basically. So, you can see that one also. And then we have to characterize them, because we have to characterize them that it is really. So, what happened? Characterization means there is no DNA, there should not be any protein, should contain only those who are scaffold related or the structure, that is all. So, here is basically, here is that beta actin is not there in this one, decellularized. What is the fibronectin, collagen 2, collagen 1 and laminin, and these are the three specific protein which makes the architect of the cardiac cells. Second thing is that images, what is that it should not have any DNA molecule. So, ethidium bromide staining, you can use that and prove that there is no gel, this is completely decellularized, there is no protein is there. The second is that in this one, that cardiogel is looks like this basically here, and then you have to do that, it helps in cardiomyogenic potential. Matlab is this cardiogel what you have isolated, it should help in, if you add any cell, just like we got a mesenchymal stem cell, okay, we have isolate from the bone marrow, okay, that I will tell you later on also how to isolate them, and then we put on them and then characterize them that yes, this cardiogel helps in the cardiac differentiation, and then you characterize with them also. All these are through the microscopically, all the antibodies and also molecular biology, you can send them, there they are there. So, these are here basically cardiac actin is there, cardiac troponin and also GATA4 and also another markers. These are all 4 markers, they are the hallmarks of the cardiac differentiation. So, you prove them, they are there, and then also you do the, you analyze them through the RT-PCR also real time, that it is there in that one. Then also you do that the angiogenesis is one of the things, because the blood has to supply in those areas. So, you see the cardiogel helps promote the angiogenesis. So, this is basically control, and then you can see the experimentally also they are also making a tubular tubes in that one. So, you also prove that one, and here is basically you can see the architect of this looks like a they are the cardiac tissues, cardiomyocyte. So, you prove with this thing. So, basically what we have done basically in summary, the cardiogels direct differentiation of bone marrow cell into the cardiac things and also basically angiogenesis, and two important things for tissues that you have to do that. And these stable clones, they are, when we express, we can track these cells when you put a GFP label mesenchymal stem cell, we can track into the cardiac cell also, that they are coming from the same pool, they are not coming from the other sources also, that you should also prove that. So, now we have to develop the patches. The patch was done basically electrospinning basically in this one. So, what we have, synthesis of molecules in this one, and we characterize by the same, we characterize, this is the basically bioadhesive, I will tell you about that. This is we have patented how we do that. So, we are not going to tell lot of things about this, how we develop. These are chemical based basically fusion of two different chemicals which are known chemicals, but the ratio was very different. So, it took us approximately 1 year to find out that bioadhesive really helps in two things, adhering to the tissues, the ventricle, and second thing is that it allows cells to migrate from the patch into the tissues, that also there. So, this is a basically characterization of those patches, how we normally we do that, and there also the quantitation of this all these cells, how many cells are converted in those things. Okay. So, this is a normal practice of making a scaffold basically and characterization. So, bioadhesive and biocompatible means whatever bioadhesive is there, it is not toxic to the cells, that is also we have to look. So, we have to look about the cell proliferation, we have to look about this longevity of these cells, how long they can stay there in bioadhesive also is very important. So, this we have characterized in this one. So, now we have to see about the morphology and assessment, how it looks like when it become a myocardial infection. So, this work was in collaboration with the CDRI Lucknow because we do not have a facility to develop a MI model, because you have to develop MI model and they have the capacity to do that, facility to develop MI model. So, we develop in collaboration with those things and we characterize this infarcted region has this kind of region in this one to histology, and we also see that how much it looks like in this one when we do that. So, what happened basically, two things we have to do these things in a controlled way, because to prove anything we need to have a proper control. So, we took only the bioadhesive and cells only, we did not put anything else. So, we see that the cells are passing through. Second thing, the bioadhesive are put in the cardiogel and the cells are embedded into that one and put into those things, and you can see the difference here. Whatever there was there in this one, it is stored here, restored in the, looking just morphology, and then we characterize in all those things how much cells are integrated into the area, because they are GFP labeled in that one. So, that is why we able to confirm that whatever restoration happens by the our cells only. So, and then you have to do some physiological assessment of those things, just like it with the ECG. So, they have a machine to develop to see the ECG of the mice, or we can, these are basically in big rat we have done, because the mice heart is very small. So, we have to take the rat only, there we can do that. So, after assessment what happened, the two things are, ejection volume is the one thing is very important. When you see the ECG, how much the blood is ejected when they are contracted, and that shows that how much capacity of these cells have it to pump this into the lungs basically for purification. Okay. So, that is the one criteria they use, doctor use to find out whether the heart is working properly or not working properly. So, that is what here is that. If you see here, I think this is not clear to you, but I am sorry for that, this is a little bit different, but here is very clear in this one. So, here is that, if you see here, the ejection volume is here, this is the control, and you can see the volume of ejection volume is almost to the control here, which is not 100% same. This is a control without MI, and this is MI which is reduced here, and these are different condition 8 hours, 24 hours and 48 hours after the adhesing the patches. And you can see how this significant functional is increased here in this one, if you see here with the control. So, we have done the cardiac functional parameters and histological parameter. We can say, if you see here, this is almost the same basically here in this one, control versus this one, after 48 hours of transplantation of these cardiac patches. And these cardiac patches are basically only 5 millimeter, not more than that. What we are thinking that if this is a proof of concept we have done, and we have published this paper also in this one, what we are thinking that if this is working, so we have to go for a little bit sheep model, because there we can increase the size of the patches. Here all the bone marrow cells is taken by the same mice, same rat. We have not taken anything from outside. So there is no problem of accepting those MSCs, although MSCs are basically immunologically safe. I mean, you can put any other mice into that one. That's what the MSC transplantation is much better in that one, because they don't have any adverse effect. They don't develop immunogenicity when you use the same thing. So MSCs are immunocompromised cells basically in that one. So now what happened? This is okay, good, but the problem is that we have to increase the functionality of the differentiation process. How we do that? Because in this I have not used anything extra. What you have used only the cellular components to convert into that one, and whatever the agents are, the enhancer is available in the vicinity of that particular ventricle only. So can we add azacitidine, which is known differentiation material, into that one? So what we see when we want delivery of these things into that one, either we can add here. So what happened? We have encapsulated in a nanoparticle those azacitidine. For that we have not used any chemicals making those nanoparticles. So what we did, so this is summary of those things, the last things. The next thing was that characterization nanoparticle, and everybody does nanoparticle. We have used basically a zine, which is basically plant product, okay, and this is basically recyclable. It is not this has been used for several other purpose also, but nobody used for cardiac regeneration. So we have used this for delivery system. We have made a nanoparticle, we have characterized according to morphologically, TEM, SEM, FTR, IR, all those things, whether what are the functional groups are there, it is attaching or not there. I think everybody knows that. These are all conventional things, you know. So 5-azacitidine gene nanoparticle synthesized and shows sustained release of here of azacitidine, because we want that if the patch is stays for 48 to 96 hours, and these patches are biodegradable basically, which I forgot to tell you, because this is coming from the biological material. So they have actually in 3 months you don't find any scar, you don't see anything available in that one, but the drug has to release because cell has to be differentiated. So we need to have sustained release here also. So you see 96 hours slow release is there. So continuously azacitidine available for differentiating process purpose in that one. So we have this one, the release of, so this nanoparticle is good. Now we have to incorporate these nanoparticle into the patches also. Same thing. This is hydrogel, how we characterize this, everything is there, all these things are there. This is all material. Where is this engineering department, rheological study, swelling behavior, all this physiological or biological characterization we have performed here. And then we had their biocompatibility, that when you add this into that one, it should not destroy your cells. Okay, so that we have done everything. These are basically distribution of those things here, characterization of this. Basically, this is different technologies. This is just like computational things are there, where you can see a very nice images where you can say that they are working fine in this. And gene expression analysis, that how much genes when you add this molecule into those MSCs, they have to differentiate. So we have to prove that yes, these are making these MSCs into the cardiac cells. Second thing is that there is a viability test there, they are not destroying, but they help in proliferation. So that's what this shows, that they help in proliferation of stem cell first and then differentiated. because any cell has to proliferate to and then differentiate. If they differentiate, then what happen? The number of cells which are differentiated will be always lesser and lesser. So, they need to proliferate first and then differentiate. So, enough cells will be differentiated into the cardiac cells. And then we have to in vitro vivo assessment of the hydrogels, that it is there, and you can see here, if you see in this morphology here, this is angiogenesis which is increased with this one. So, it is not hampering all the properties which I talked earlier, except it is increasing the potential of the stem cell converting to the cardiac cells. And then we perform on the same kind of ejection volume and all the parameters in this one, because we have to physiologically we have to characterize them, that it is not hampering anything in this one. So, you make in vivo assessment, again same thing, we develop a patches, we put patches on the rat and then we did it, similar kind of thing, and you can see the ejection volume is less compared to this one. So, here is all these parameters are done by through ECG. So, you require basically a veterinarian doctor to help you to understand this process, and we cannot do that, and that is why CDRI is very helpful, because they have veterinarian doctor who can help me in reading all these things with me. So, significant functional improvement in the cardiac parameters and attenuation of the adverse cardiac remodeling. So, if you see the patch is biodegradable, number one, it does not stay more than 3 months. Within a 96 hour, there is a 17 to 20% increase in ejection fraction, which is a significant, and after 3 weeks in rat, I am not saying in human, in rat we can see a complete remodeling of those scarred tissues. So, in summary, we can say that synthesis of gene nanoparticles, synthesis of characterization, injectable hydrogel, evaluation of in vitro evaluation of injectable hydrogels, in vivo assessment also we have done, and is very good. So, in conclusion, that our finding demonstrate in promising approach to repair the ischemic myocardium through tissue engineering and the cardiac patch and hydrogels. Basically, in future we have to do large animal studies for evaluating this translation efficacy of the cardiac patch and injectable hydrogels. If you have any question, I can answer. Thank you. Yes, please.
Thank you so much for interesting talk, sir. Actually, I just wanted to know, does the size of the cardiac patch matters?
Yes, because if you put too much cardiac patches, because it will also cover the which is not infarcted region. So, you have to put only that particular area or less than that, because you cover that, you are hindering the functionality of the right tissues, which is functional.
Drug enclosed
see if you see these nanogels are incorporating the patches in cardiogel itself. So, there is a cardiogel is there, matrix, stem cell are inside there, and this outside these nanoparticles are there. If these sustainable release into the vicinity, and then cardiac cell MSC differentiated in cardiac cells. That is basically. So, if you put too much azacitidine, that is also not good. So, you have to standardize the situation where the maximum conversion of these MSCs into the cardiomyocyte tissues is required, I mean observed. Yeah. Known. Yes, that is how it increase the differentiation. You are right. It is a demethylizing. Genes, right, correct. We have also published that one, that what are the gene involved in that one. Yes, yeah. We know all the inside. Yes, epigenetic genes are very important. Yeah, so not only the inside, but epigenetics are much more influential in developing the tissue regeneration. It is much important in that one. Yes, yes.
then you told that you got secondary cell culture, like differentiating, or you just use the primary?
Primary culture, no secondary culture. 3 or 4 passage maximum.
Because chances of differentiation is high.
Yes. So, because what happened, the moment we take after one prakash, we add some decellularized material.
I'm curious to know what is the size of heart then just one day mouse.
Oh, just very small, yeah, tiny bit, yeah. But you get lot of cells. You require 6 or 7 unit mice to do the studies, yeah, minimum. Yeah, please. See, when they proliferate in cardiac tissue, the final differentiated tissue, after that they do not differentiate, they do not proliferate at all. It is only when the final de-differentiation is different thing, it is only differentiation. So, the differentiated tissue do not come back to the cellular stage, it become differentiated and it stays there. Yeah, there is no backward, just like in our development process, when the cell divide, it has a destiny to form certain tissues, right, and the tissue does not come into the cellular components, whereas the stem cell has the capability to form and then stay there, it will not come back to that thing. Yeah. So, what happened, this scaffold is there, which is basically cardiogel, it is a very it is kind of gel, okay. Then you have to put a bioadhesive on top, bottom of that thing. The moment you bioadhesive, the cell attached, the cardiogel attached to those, and this cardiogel containing the cellular component also, and then they are adhesive, and then you put immediately onto the infarcted region. So, first of all we have to look around that this infarcted region attached with those cardiogel. Its attachment is not proper because the heart is continuously beating. If they are beating, if the adhesion is worked, we use basically glue, surgical glue before. When we use surgical glue, the cells are not passing through. Huh? Yeah. So, we could not see any movement of, that is why we use the GFP level MSCs, that they have to pass through. So, we could not see that, and it become attached in such a way that it is very difficult to take it out also. It was not biodegradable also in that particular. So, the surgical glue which are available in the market, surgical people use it in hospital, was not helpful. So, that is what we went and developed our own adhesive using different chemicals, and these chemicals, because has to be in such a way that should not be toxic to the cell also. So, we check all the toxicity assays in the tissues first, and I know you guys know how to do the toxic assay in live animals. So, we have performed those things after that. This work took around 5 years to come at 2 papers only. But the reason, so far out of 10, 8 were successful. That is what we did. Otherwise, corona came and we did not perform more tests. And that is what the problem is, that as the number is more, you are much more confident. You can do that. For lead examination, what is the meniscus is there. So, what we have done, I think that is also we have developed a 3D printing and bioprinting. So, we have printed the whole material with cardiogel along with the cells, and we are thinking that we will do that, okay. But meniscus we have printed and we published the paper also. In that one we developed the scaffold, we published the meniscus and we put in those things. We have not done in vivo studies yet, but in vitro we have done it. What is the toxicity, what is the viability, survival, everything we have done it. But unfortunately, those 2 people who start working, they went for postdoc. It's a sustained release, the same thing if you see that, yeah. No, pH is basically is not very high here, okay. There is no low or high pH in the something neutral kind of things is there, yes. Yes.
So instead of putting a biopatch, it's just inquisitiveness. Instead of putting a biopatch, can we just allow the
See, the
targeting
number of cell where reaches in destination
Right.
is very less.
In
How do you direct that they should reach only that cardiac patches?
some trafficking marker, those may be in the hypoxic condition or something?
People have tried injecting those cells in the particular vicinity also, but the number of cell stays, that is the beginning I told me, it's a problem there because the cells are always waiting. So there is a fluid is there and they goes out of that cell. They will not start the function. They have to stay few hours there.
Right. In your opinion,
Yes. Oh, not open heart, you cut it, open it and put it and seal it, yes. Just one time surgery, that is what we are thinking. So we are thinking that if this happened in the sheep model, we can develop basically a tubular surgery and put on top of those things and put there immediately. So you do not have to open the whole heart. So make a little bit of hole, not laser, what you call it this? Laparoscopy, yes. With laparoscopy surgery, if you can able to do that, because it is much easier, because we do not have to whole open heart, you have to find the particular place. If you open little bit and put it there, that is enough for that. Yeah. No, but we have to remove the upper layer because heart is also covered with the, so we have to little bit open that and then you put it, otherwise it will not go in. So these are all practical problems are there which we have to solve it. Unfortunately, when I am the peak, I am retired. Yes. No, cardiogel was not developed in spinogenic electro, this one. No electro. Gel form. See, when you do the electro spinning, yes, yeah, chitosan, yeah, yes. There the lot of studies are there. Yeah, they do convert into the cells into the cardiomyocytes, but how to develop the delivery point is a problem, because if you see the chitosan, it has basically more than 6 months of biodegradability in that one, where this cardiogel has within 3 months it is whole biodegradable. Even you can see after 3 weeks 60% is dissolved in that one. We have tried those things, but again if you do not put any adhesive, because we require still adhesive require, but if the patch is like a dermal patches, it stays in particular area and also delivery is there. So sustained delivery of the molecule, which is azacitidine we have talked about, and also the cell has to be crossover. See, here in dermal patches only the drug has to cross, nothing else, right? Here in those the cellular component which is much more larger than the drug, if you see the whole size, they have to cross. So the gel has to be permeable to those things, that is also very important. And sometimes chitosan become, if you electric spin them, they become very tight, and the gaps in the porous form, we need a porosity in that one, so that and then hydrophilicity also, these two things are required, and cardiogel has both in that one.
Do we have any more questions? So thank you, sir, for the interesting talk.
Thank you.
We also have few questions from the participants who have joined us
Please.
online.
Yeah, okay.
So I'll read it on behalf. I have a question from Dr. Debate, and he says that with regards to fabrication of different drugs, what challenges is an approval from the competent authorities like FDA and
Yes, it's a very good question, because what happened, first you have to take ethical clearance, second you have to take animal ethical clearance, okay, and that took a long time. First thing, second thing, if you are not doing in your institution, you have to take other institution ethical clearance and ethical clearance in both. Since it is not human cell, FDA will also do a lot of scrutiny in terms of how these are helpful in the human being. When you talk about the human, we need to have sheep forms. Then again FDA will also require to see all those they do a toxicity, cell toxicity is very important, survival, those things, and second thing, how effective these are, efficacy. These three things they will look in a great detail, and number of the animals and number of the experiment, because you have to have a statistical significance. So that also you require more. You can't do a one animal study and get the approval. It's not possible. You need to have a, at least 10 experiment in the sheep, and then maybe monkey or dog, and then it will go to the human. So it's a long process. That's what happen in drug development. It takes around 8 to 9 years because of these things. I hope I have answered this question.
Thank you so much, sir.
Any other question?